Hello, everyone. I'd like to welcome you to our talk today, both of you that are currently with us live and for those of you that are watching this as a recording. The title of today's talk is So Many Procedures, So Little Time, How to Develop an Effective Schedule of Events for the Rare Disease Clinical Research Network. To give an overview of the presentation today, we'll have a brief introduction of myself and my fellow speakers. We're going to cover some of the resources available that have been created by the DMCC for members of the consortia. And then we're going to get into the bulk of our talk, which is going to be on what we call the 4 pillars of an effective schedule of events. And we're going to kind of give a demo on how to create a schedule of events and some common issues that arise when developing schedule of events for human research subject protocols. And then at the end, we'll have some time left over for some questions that hopefully we can answer anything that arises during this talk. To start out with introductions, my name is Mike Fusacchio. I have experience both in the contract research organization and academic regulatory environments, where I've led the submission of both IND and NDA projects to the FDA.
My name is Nathan Hawk. I'm a senior data specialist with DMCC. I have over 14 years of data management experience, 6 years in the CRO, 8 years in academic research. I have experience with drug and device trials as well as general clinical research projects.
My name is Sam Sonoda. I work at CCHMC as a monitoring lead for our investigator-initiated studies, and I previously worked at a CRO in a monitoring capacity as well.
Hi, I'm Lisa Tully. I have over 15 years experience in academic regulatory research here at CCHMC. I have over 10 years as a clinical research coordinator and more recently with managing regulatory for multi-site studies as well as IND projects.
Great. Quickly, just wanted to kind of highlight some of the resources the DMCC has made available to consortium members. The first is the protocol template and checklist. So these are 2 separate documents. The checklist details all the sections and potential subsections that you could have in a protocol to allow you to kind of decide what items are necessary when you're kind of creating your protocol. The template provides those sections and subsections along with some standard language to assist in writing certain protocol sections. This is both just kind of standard language and standard language that the DMCC would prefer to be in those sections. Based on feedback from consortium members, we created the pilot project protocol template. This is ideally for smaller initial studies or studies that involve only human samples or existing data. It's based on the standard protocol template, but has been significantly trimmed down in regard to which sections are present to make it more appropriate for these types of projects. Because a lot of the documents that we see in the DMCC are initially coming from grants, we created a grant to protocol conversion document that highlights some of the key differences between a grant application and a human subject research protocol and how to go about converting those sections. And then one of our newer documents is the Manual of Procedures template. This was created to aid in constructing an effective manual of procedures to aid each study team in their upcoming trials. So, schedule events are a very complex portion of your study. They require a large amount of input from everyone that's involved in your study team. That's going to be your PIs and your sub-PIs, your coordinators who might be doing individual visits, the data management team who's going to be handling the data, monitors who will be reviewing how the different visits are going and making sure that subjects are visiting under safe conditions, statisticians to make sure that everything is being evaluated in a way that meets the statistical analysis requirements of the study. So as you can see that an effective schedule of events really requires a large amount of input for something that is a smaller portion of the protocol, but in all honesty, really kind of represents the protocol as a whole. And so what we've created here is an example of an effective schedule of events. And so throughout this talk today, we're going to start at a much more basic version of this schedule of events and build towards this schedule of events by addressing common issues that arise when teams put together a schedule of events. So when putting together this talk, we really kind of came up with the idea that there are 4 pillars of an effective schedule of events. These 4 pillars are your study design, the workflow, data management, and quality assurance. An effective schedule of events will properly address all 4 items and properly set them up. And that appropriate foundation for that schedule of events will allow it to be used effectively and your protocol to move forward more smoothly and help alleviate any potential negative outcomes from something being missed or something not being done at the correct time. So we're, like you can see here, they're like the columns or pillars of the building. If you remove one, you run the risk of your building collapsing, or in our case, of your study not progressing appropriately. So our first pillar, study design. So how does the scientific rationale of your potential study affect the schedule of events? You really need to look at what we kind of consider 3 key sources of inputs regarding scientific rationale when you're creating your schedule of events. The first is your standard study events. These are elements that should be a part of every study. You have informed consent, inclusion-exclusion criteria, your medical history, the demographics, data that you're going to want to collect, or procedures that have to occur in order to run a human subject research protocol. The second is your standard safety events. These are your safety elements that should be part of every study. Reviewing adverse events, concomitant medications. These things should also probably be done at every visit. Especially with the concomitant medications, because you want to be able to see, has something changed in between visit 1 and visit 2 to ensure that you have the most up-to-date data when you're assessing any potential adverse events. And the third, and really an important factor, is the scientific rationale itself. You should always review your schedule of events versus scientific rationale for that trial. Are the tests and procedures that are listed in the schedule of events able to represent all the endpoints for that study? If you look at the schedule of events, can you actually answer and obtain the data you need to address your endpoints? But at the same time, you want to look at them for, to make the schedule of events easier to use. Are there procedures that can encompass multiple elements? We can use the term blood chemistry, but when you use terms like that or rows like that in the schedule of events, you need to add the appropriate footnote. You want to make sure that anyone using the schedule of events understands what that procedure represents. So you need to specify then through the footnote exactly what blood chemistry means. And then with the rationale too is, when will this information or these samples be collected? If part of your endpoints is looking at how a particular biomarker changes throughout the course of the study, you need to make sure your schedule of events shows that you're going to be collecting that biomarker or the necessary way of collecting that data throughout the study at the necessary time points to address your scientific question for that study. So as I kind of mentioned earlier, we're going to take a very basic version of the schedule of events we previously introduced and kind of develop it as you might develop a schedule of events when you're coming up with your human subject research protocols. So we're going to take this basic version of the schedule of events, and we're going to kind of review the 4 pillars throughout this talk and kind of address some common issues that are made regarding those 4 topics. So for this practice example, we're going to consider a trial that has the following parameters. There's going to be 4 visits, screening, day 0, day 7, and end of treatment. It'll have a placebo and a treatment arm. The study is looking at whether a drug reduces the level of triglycerides. This is going to be considered its primary endpoint. And there'll be several laboratory procedures, an ECG, some blood draws, pregnancy tests that are for the purpose of either inclusion-exclusion criteria or potential safety efficacy endpoints. So you can see here we have a very basic version of a schedule of events. But as you kind of review this slide, you may start to see some issues that are present in this schedule of events. So throughout the rest of this talk, we're going to start to address those potential issues, starting with those relevant to study design. So in regards to study design, there are really 2 major issues that we want to highlight initially. The first is that, as we discussed, we really should include some of these common study safety and study event items. So informed consent, adverse events, medical history, concomitant medications. None of them are currently listed in the schedule of events, and so we want to add those. The second content issue is that it's unclear if the primary endpoint is being evaluated based on the information provided. So you can see that we say labs on the row, but does labs mean that triglycerides are being evaluated? We don't really have a way of being sure of that based on the way the schedule of events is currently shown. So you can see here now, we've updated the schedule. We've added, in regards to the first item, we've added informed consent, medical history, review of concomitant medications, inclusion-exclusion criteria being evaluated on the first day, review of adverse events. For the second issue, you can see that we've added a footnote to the labs row and included the footnote that say labs will include a CBC and a lipid panel. So we now know that triglycerides will be measured on all 4 visits. And so we're now more confident of the fact that we are evaluating the primary endpoint at every study visit. So now we're going to move from the first pillar onto the second.
Thanks, Mike. So as Mike said, our second pillar is workflow. So a question I want you to keep in the back of your mind is, does the schedule of events accurately reflect the protocol workflow? As you think about that, let's look at 3 key elements that are important to a solid schedule of events. Those are clear, consistent, and concise. Being clear means a study coordinator can run the study visit and procedures correctly, completely, and consistently each time he or she conducts a study visit. It also means each coordinator will run the visits and procedures the same way, reducing variations that could impact the collection of data and thus the results of the study. Some examples of what should be clearly illustrated in your schedule of events include: 1, the duration of the study for each individual participant. Will they be participating in 2 visits or 10 visits? Will they be in the study for 2 weeks or 2 years? It's helpful to have this visual timeline within your schedule of events. A second example are visit windows. Do visits have to happen within a tight timeline, such as scheduling visits within 2 days of the previous visit? Or do you have some more wiggle room to schedule your visits, something more like 3 weeks in which to schedule the visit? A third example are procedures. What procedures are to be completed during your study? Are they clearly listed out? Are each of the procedures to be completed at each study visit, or do the procedures vary at each visit? So it really must be clear which procedures are to be done at each visit. Something else to consider are questionnaires. Do you have 5 questionnaires or 10 at each visit? When creating your schedule of events, think about listing maybe just questionnaires on one line, and then in a footnote, list out the names of the questionnaires to be completed. This could reduce the number of lines within your table while still giving the necessary information. Our second key element is consistency. What is written within the body of your protocol must mirror the schedule of events. So if you're using the RDCRN protocol template, this specifically is section 7, study procedures. And examples for consistency are, if procedures are to be performed in a set order, make sure it is clear what that order must be. Maybe even list the procedures within the schedule of events in that order. Also, is a change in the order allowed? Make that known in the protocol text in that section 7, but also make a footnote in your schedule of events. Something else to aid consistency is to think about what is included in the various procedures. If the protocol states a physical exam is to be performed at the baseline visit, but an abbreviated exam will be done at other visits, what's the difference between those two exams? Maybe list in the table just exam, but then add a footnote to explain what will be done for each type of exam performed. This could help with ensuring the correct exam was completed at the correct time point in the study visits. A third example are the terms used consistent between the study procedures and your schedule of events table. Make sure visit 1 is listed as visit 1 in both the text and your table. Don't have the study procedures state baseline visit and then your table state visit 1. Being consistent will aid coordinators in completing their visits correctly and will also help with discussing the different visits with participants and their families. Another item to think about is whether the schedule of events used in the protocol matches what is used in the consent form. You have to ensure they are consistent. It will help participants be fully prepared for what will occur during a visit, and that way they won't be met with any surprises because something additional was listed in your protocol schedule of events, but not added to the consent itself. The third key element is concise. This is finding a balance between adding in all necessary information, but keeping your schedule of events readable and understandable. If too many items are listed in the table with, for example, the full names of each questionnaire, it can appear cluttered and confusing, especially if your spacing truncates the names. However, too little information will lead to coordinators potentially performing study visits or procedures differently, or maybe even forgetting something and missing data. So it's really a balance. Do you have a lot of abbreviations? Maybe think about listing the abbreviations in the table and defining them in the footnotes. Maybe do you have a large number of footnotes and a large number of abbreviations? Then consider creating a footnote to indicate all abbreviations are defined within your abbreviations table in the beginning of your protocol. Then I would recommend ensuring all your abbreviations are actually listed in that table. This could help reduce the number of footnotes and help ensure other necessary footnotes aren't overlooked. Taking the example of procedures again and the different types of exams. Do you have a complete physical exam, neurological exam, and abbreviated exam in your study? Are they all completed at each visit or at different visits? Does it make more sense to list each exam on its own line in the table with a description of what is included within your footnote? Or is it more clear to simply list exam in the table, but then in the footnote explain which exam will be performed at which visit? An additional consideration is if one or more exams will be a part of your endpoints, then you will definitely want to list them separately to ensure they're completed properly. And lastly, think about your study-specific details, such as which color blood collection tube to use, or in order to take a blood pressure, the person must sit for 5 minutes and then take it twice. Those are types of specific instructions or procedures that are generally better suited to be placed in a manual of procedures. So back to our initial question, does the schedule of events accurately reflect the protocol workflow? If the schedule of events is not clear, consistent, and concise, it can lead to a protocol deviation if a procedure or assessment is not completed or completed incorrectly. It can also lead to missed data if something is overlooked, all while potentially impacting your participant safety. So let's take a look at our example schedule of events again. When scheduling, as coordinators know, we need to schedule the study visits according to the protocol, and if we don't, we'll have a protocol deviation. However, scheduling with participants, especially kids, can be difficult, working around school and their other activities. What can help us out, though, is if we build in a cushion of time with our visit windows. Also, looking at this as it currently is, we don't know how much time should be between the visits. Should they be weeks apart or months? Adding both the time needed between visits and windows in which to schedule is necessary to ensure everyone follows the same time frames. The other issue in this example is that it doesn't specify what is included in a physical exam. Sometimes people consider vitals, height, and weight to be part of the exam. Sometimes studies use a complete exam and an abbreviated exam. So what's the difference between the two types? So here you can see, as we added in the necessary time between visits, along with our windows for scheduling, it aided our clarity and consistency and eliminated our first workflow issue. So you can see screening takes place 30 days before day 0, and the end of treatment visit should take place 30 days after day 7. Then we have the additional visit windows of either 5 days or 1 day. For the physical exam, by including a footnote, we concisely explain what will be included so everyone on the research team is aware and conducts the exam, including all necessary elements. In our example, the exam includes height, weight, vitals, a musculoskeletal assessment, as well as a cardiovascular component. Also note that as we updated the footnotes, we put them in order rather than leaving them jump around. So you can see physical exam is now footnote 1. So with that, I will hand it over to Nathan as he looks at our third pillar, data management.
So for the third pillar, data management, the following attributes of a schedule of events are essential for execution of data management responsibilities. We look at structure, we consider the order, the granularity, and the context of the schedule of events when we begin to design the database. So structure is very important. It's very important to have all procedures that are represented in the schedule of events as they are described in the body of the protocol. As previously mentioned, it's important not to forget those common data elements such as informed consent, eligibility, randomization, and demographics. A basic rule is if you expect the data to be entered in the database and used for analysis, it should be listed in the schedule of events. If it's not, you run the risk of it not being included in the database and not being available for an analysis. As data managers, one of the first places we go in a protocol to begin to think about the database build is the schedule of events matrix. It's a really great visual representation of how we would potentially spec a database build how we communicate to our programmers for that build. It also is a really good visual representation of how some databases look to users. So it's really nice to see that matrix to get an understanding of, at a high level, what procedures are necessary, what events are included for a particular protocol. Order is the next important point for data management. It's important that all procedures are in order and consistent with how they will be done in real time at each visit and event. Some examples would include, if timing of procedures is an important element for the protocol, having the forms listed as they occur chronologically will help ensure data collection is done correctly. Databases are typically built with forms ordered as they appear in the schedule of events. Listing the procedures in an order consistent with how they will be done at the visit will make data entry more efficient for site personnel. If the order of procedures or forms in the schedule of events does not match the rest of the protocol, it may create confusion for the person building the database. That confusion ultimately leads to wasted time, money, and/or inaccurate database builds. And the order is also important for data management when we think about data quality and the data cleaning checks that we will implement in the database. So knowing the order and the timing will help us set up accurately what data should be collected in what order, and so we can program queries to auto query in the system. Granularity is the third consideration for data management. Line items should be represented in logical domains. It should not combine differing concepts or procedures. So when I say domains, the term is derived from some data management standards called CDISC and CDASH. Demographics would be a domain, adverse events would be a domain, concomitant medications would be a domain. So when I say domains, they're typically concepts, differing concepts. So we want to avoid combining concepts that are different on line items just to clearly communicate what needs to be built. So one that's commonly, domains that are commonly combined are demographics, medical history, family history, and medication history. They might get combined under medical history. It might be better to list those out as line items because they are differing concepts. And another example would be echo, CT, MRI. They're all imaging. They could be labeled under imaging, and we might not know what type of imaging needs to be done and what forms need to be built relative to that line item. As an alternative, it's okay to have one line item if you have a footnote to provide and to define what domains and procedures are done as part of that line item in the schedule of events. It's okay to combine them to save space. Just be sure that you clearly identify what procedures are done and not done at each visit and event. A good example of a line item that can be combined would be labs. You know, you have subcategories of labs, for instance, blood or urinalysis. You don't need to list blood and urinalysis as one or as separate line items. It's okay to list it just as a singular line item under labs, and then put in the footnote that you're going to be doing blood and urinalysis, just so you have that granularity and we know that there are different types of labs being done. The fourth element for data management is context. All context as applicable should be described in the schedule of events. Some examples include timing of events and procedures should be clearly defined in the schedule of events. If vital signs should be done 30 minutes prior to study drug administration, the schedule of events should note that timing. As previously mentioned, defining visit windows in the schedule of events is very important. Again, that will impact our data quality. So we would be checking that the data was collected at visits per the protocol timing and windows established. If there are multiple types of procedures, specify what kind. Is it a resting or stress ECG? What version of a questionnaire should be administered? There can be multiple versions. Is it arterial or cuff blood pressure measurement? Failing to provide this context can lead to inaccurate, mistimed, and or incomplete data collection. To go back to our schedule of events example, to highlight a few common data management issues, we have an example of a granularity issue and a context issue. The first issue highlighted is when we initially addressed the content issue regarding the study endpoints, we created a data management issue by not specifying all the labs that will occur. So if you see the lab note, it says that it will include CBC and a lipid panel. The second data management issue, the schedule of events calls for an ECG, but provides no information on the ECG. Is it a resting ECG? Is it done in triplicate? We don't really know because we don't have that level of detail. As a correction to the example schedule of events, we've added a note to labs to indicate that we will be doing a CBC, a comprehensive metabolic panel, and a lipid panel. So previously, the comprehensive metabolic panel was not included in the footnote, would not have been built by data management. So it's important to highlight that granularity. The ECG will occur after the participant has been resting for 5 minutes, and it will be done in triplicate. So also important notes for data managers to know what variables need to be built into the database, and also any sort of timing elements that need to be programmed for data quality purposes. And now we'll move on to the fourth pillar with Sam.
Thanks, Nate. So for the fourth pillar of quality assurance, I wanted to set the stage and start off by outlining 3 key elements to quality assurance as influenced by GCP. So these include subject safety, study quality and integrity, and compliance. For subject safety, we want to ensure that the rights and well-being of subjects are protected. For study quality and integrity, we're responsible for ensuring that data is accurate, complete, and verifiable from source. And we also want to develop a protocol that provides a consistent and quality approach to the conduct of research among investigative sites. And for compliance, we're ensuring that the conduct of the trial is in compliance with the protocol, GCP, and any applicable regs, whether they be local, federal, or institutional. So with these key purposes in mind, I'm going to dive a little deeper into each element and then focus on how we can view creating an effective schedule of events through a quality assurance lens. So starting off with subject safety, the main question we want to consider is, are all safety procedures conducted at the appropriate time points? So here we have a list of examples for common safety procedures that are done throughout trials. And depending on the patient population, the risks of the study, and any eligibility requirements, we want to make sure we're adequately monitoring subjects throughout their study participation and ultimately enrolling the desired patient population. For interventional studies, we also want to review the IB or package insert to ensure that the protocol procedures align with any recommended safety guidance as well. So when it comes to study quality and integrity, it's crucial that we take a look at the big picture and ask ourselves, do the procedures and SOE exhibit a consistent and quality approach to the conduct of research among investigative sites? Meaning proper performance is outlined, especially with regards to the objectives and endpoints of the study. And if the study is multi-site, the procedures should be listed as applicable for all sites or noted otherwise. Lastly, for compliance, study teams should review the protocol from an outsider's perspective. So the main question we want to ask from a compliance perspective is, would the procedural expectations be clear in the eyes of a monitor or auditor? So we've touched on this throughout the presentation, just to keep in mind the perspectives of a coordinator or a data manager, but just reiterating that timing and specifics on procedures should be clearly defined, and important details that may not fit within the SOE can be provided within the footnotes. Lastly, another important consideration with compliance is that informed consent is conducted prior to study participation. So we want to ensure that ICF is listed appropriately within the SOE as well. So the two key takeaways I have for quality assurance are, one, that the SOE should be comprehensive and allow for the study team to implement and maintain subject safety, study quality and integrity, and compliance. And two, that studies teams should review the SOA through a quality assurance lens to do two things. The first being identify potential issues that may occur due to infeasibility, and then also identify potential issues that may occur due to inconsistencies across the study documents. So with that, we're going to move forward with some examples of common QA issues within the SOE example. So the first example we have of a common QA issue is the pregnancy test collection. So if you take a look at the schedule of events here, this is a potential issue that can occur due to inconsistency. So our example protocol requires that at the screening visit or during IE evaluation, we are looking at pregnancy status. So we need a pregnancy test done at visit 1 or screening, but we don't have a corresponding X in the table. Then the second issue occurs within the footnotes for the requirement of the ECG. So here we have that the ECG will occur after the participant has been resting for 5 minutes and done in triplicate. And this is an example of a potential issue that might occur due to infeasibility. So essentially, we're creating a situation where getting documentation that a subject noted rested, that they rested for exactly 5 minutes may be unlikely or inaccurate. So we should adjust the wording for that footnote. So in the second slide, we've addressed these two issues. We've added a PT to be done at visit 1 or screening. And then we've added the term approximately to the footnote to give some leeway for collection of ECG and to prevent protocol deviations.
Thanks, Sam, and everyone else for kind of going through their individual sections so far. So what we really wanted to highlight today is that the schedule of events is a very comprehensive item within the protocol. And you can see the schedule of events that we've listed here on this slide. It's the same one from the beginning of the presentation. Is obviously a very considerably better version of the schedule of events for this potential protocol than the one that was kind of our initial starting point. But I think the key thing to take away from this is that there's still room for improvement. You know, as kind of Lisa pointed out, you know, we use some abbreviations in this schedule of events, but we don't necessarily have them defined within the table. You know, other things to consider: if this study had a early termination visit, how do we have that represented in the schedule of events? So due to the complexity of schedule of events, it obviously requires a large amount of input from your own team and everyone that's going to potentially be involved in the upcoming trial. But obviously, you know, it's the more eyes you can get on the protocol, the more eyes you can get on the schedule of events. There's obviously, you know, better off for making sure everything moves along smoothly. So the DMCC highly recommends that, you know, a schedule of events table be present in any protocols. That way that when we're doing our reviews, we can try to point out any issues that we notice based on things that we've reviewed and kind of hopefully address these issues early on. And that way they're not issues that are occurring after the study has been opened. So you're not having to do protocol amendments or respond to deviations, or, you know, potentially missing out on any potential data points, or, you know, even worse, having any issue concerns with patient safety. So with that, I would like to thank everyone for attending the talk today, and we will open it up now for any questions that have been raised throughout this talk. Thank you.